So welcome to California Invasive Species Action Week. This is under the direction of the California Department of Fish and Wildlife and aims to increase public awareness of invasive species issues and promote public engagement in stopping their spread. Events such as habitat restoration and invasive plant removal days take place throughout the state from June 1st through June 9th. There's a youth art contest. And for the 7th consecutive year, UC Agriculture and Natural Resources and the California Invasive Plant Council are partnering on these invasive species lunchtime talks that take place at noon daily from June 3rd through June 7th. I'm Randall Oliver. I am the statewide communications coordinator for invasive shot-hole borers, part of UC Agriculture and Natural Resources, and my partner on this call is Julie Clark, who is also with UC ANR at the Ventura Cooperative Extension offices. And Julie will be handling Q&A and chat and those types of issues for us on this call. I want to quickly interject that there are lots of organizations working on invasive species issues in California. Those include federal partners from USDA. Here we got ARS, Agricultural Research Service, as well as APHIS, the U.S. Forest Service, state agencies like the California Department of Food and Agriculture, CAL FIRE, the Invasive Species of California, and the California Invasive Species Advisory Committee, local agencies, county ag commissioners, NGOs. It's a really long list. And unfortunately, if we took the time to list them all, Brian would have no time to talk today.
And I'll just stop my own video here. So, well, thank you again. Thank you for inviting me to speak today about classical biological control of yellow starthistle in California. I prepared this talk with some help from Dr. Lincoln Smith, who is a retired colleague who really did the bulk of the work that I'm going to talk about for yellow starthistle, at least especially regarding the new weevil that everybody, or not everybody maybe, but some people have been asking about. He's retired, but he is still working with us as a volunteer, coming in to help out, and he's been an invaluable resource. There we go. So to start out, I'd just like to briefly define classical biological control. For those of you who may have heard the term before, but maybe aren't exactly sure what it refers to, classical biological control is the reduction of invasive weed populations to sub-economic levels via the release of co-evolved, host-specific, impactful natural enemies. And just to look more closely at some of these technical terms that I've used, a co-evolved natural enemy is one that has evolved feeding on the target weed in the native range and has adapted to the target plant's defenses. The term co-evolution refers to the phenomenon of maybe an insect feeding on a plant, and then the plant over time is selected for some defenses that prevent that insect from feeding, and then in turn that selects for insects that can adapt to that new defense, and it goes back and forth like this over what we refer to as evolutionary time. We ensure that a biological control agent is host-specific by rigorous testing on dozens of non-target plant species that are closely related to the target. And I'll talk a little bit more about that in a minute. We also test the candidate biocontrol agents to ensure that they will have a significant impact on reducing the target weeds populations following a release. If they don't, then you're releasing a new species that may have unforeseen effects in the environment, even if it's not really doing the job that you wanted it to. So even if an insect is highly host specific, if you can show in pre-release testing that it won't damage the target plant sufficiently to reduce populations or spread, releasing it would be an unjustified risk to the environment, as well as a waste of resources in preparing for those releases. So it's the impact of the biocontrol agent is also an important thing to test for before you make a determination about a release. And by natural enemies, we are talking about insects, mites, and pathogens mainly. Looking at host specificity in more detail, and there's Dr. Smith, we work with a list of plant species that would be at risk if our candidate biocontrol agent were released into the field. Typically, this host plant list includes economically important plant species that are related to the target and species that are native to the area of the proposed release, so local relatives of the target species. Testing occurs either with or without the target weed present to determine whether the candidate agent can survive on any non-target species and whether it prefers other species to the target. Results of these experiments are summarized and presented to representatives of more than 15 different government agencies, in addition to Canadian and Mexican agencies, who must vote unanimously in favor of the release for a permit to be issued. So it's a pretty high bar to show sufficient data to these panels to allow for a release. It's not just because we think it's a cool insect. For impact testing, controlled experiments are conducted to estimate the amount of damage that the candidate agent is able to inflict on the target plant, compared to a plant grown without being infested with that candidate agent. While serious damage in the lab, like you see here, doesn't guarantee a similar level of impact in the field, a low amount of damage in the lab under controlled conditions really doesn't bode well for the potential performance in the field. So again, these impact studies are important to make sure that when you release something into the field, that it's going to do something that you are intending, and rather than just being out there and just adding a new species to the environment that, if it's not doing its job, it may be having some other effect ecologically. So here is a basic timeline showing the sequential stages of a biocontrol research program. You start studying the biology and ecology of the target plant, the target weed species that you're interested in, both in its native range and in the invaded range here in America, for our agency at least. And then you go to the native range to explore and discover candidate biocontrol agents. Those candidates you test in the lab, both in collaborators' labs overseas in the native range, as well as here in quarantine situations. And the candidates that are testing well and shown to be very host-specific and causing damage, we develop protocols for mass rearing these biocontrol agents in the lab, so that when it comes time for releases, that we'll be able to release them in large numbers. Before we apply for permits, it may be necessary to do more quarantine and even field cage tests, very closely monitored. And we submit our application, assuming that all of the tests turn out well. We submit our application to be permitted to release the biocontrol agent. And at the time of the permit is when these agents can typically become available to land managers to start distributing them for releases, mass rearing, as I mentioned earlier. And after releases, I want to emphasize here the importance of post-release monitoring of the establishment and the impact of these biocontrol agents. And that information is very important to determine whether we need to use other strategies to make releases. Maybe the establishment is not as we had hoped, or maybe the impact is not what we had hoped, and we can try to adjust after the releases in that way, because you can't predict really how the insect really is going to do until you actually release. You can do a lot of studies in the lab, but it's nothing like the real environment. But that's what we're limited to in pre-release. And after release, of course, we can begin our restoration efforts to try to, for example, bring in native plant species to replace these invasive species that had pushed them out earlier. So a few pros and cons of classical biological control of weeds. It's environmentally safe due to the rigorous pre-release testing. So we expect minimal non-target effects, and it's safe for people and animals. No chemicals used. They are self-spreading and self-reproducing. So they're able to ideally establish and spread to areas that are relatively inaccessible, to provide sort of treatment in places even where you might not even know it needs treatment. And again, ideally, only a few releases should be, or a small number of releases should be necessary in the long run, with the self-reproducing aspect of biocontrol. They go forth and they multiply. There are low labor inputs, particularly in terms of when you get to the stage of releasing the insects. Before that, we're working hard here to test them and ensure that they're safe and effective. But afterwards, it's just a matter of going to the site you're interested in and opening up a can of insects. Biocontrol can be integrated with other methods of weed control. For example, herbicides, grazing, and other integrated weed management tools. On the con side, establishment of an agent can take time in the field. It's hard to predict. A lot of times you're sort of waiting for that perfect year when the weather is just perfect for that agent and they really explode and then take off from there. But it's hard to predict what that year will be and when it will happen. And there's not an immediate knockdown like you have with, for example, a herbicide. You reduce the weed populations to sub-economic levels, but you don't eliminate them. We're not eradicating weeds, we're controlling them. And ideally, we're getting them down to levels that they're no longer an economic or an ecological problem on the landscape. More than one biocontrol agent may be needed for a given weed. For yellow Starthistle, which I'll be talking about today, there's been half a dozen insects released already, and we're still plugging away. It's a tough weed. Silver bullets, that one agent that just does the job by itself, that's very rare. The agent establishment can vary based on the climate, on individual sites, the weed genetics, which I'll talk a little bit about later. It's, again, difficult to predict how well an agent will establish. And classical biocontrol is not applicable to every weed problem, especially, for example, native weeds that evolved here in America. They're already present with their natural enemies. You're not going to find new natural enemies for those overseas or anything. So here in Albany, California, our ARS lab, we are working on quite a few target weeds. Those that have agents available for release include yellow star thistle, which is the subject of this talk. Also tumbleweeds, arundo, Cape ivy, and water hyacinth are some weeds that we have developed biocontrol agents for, and are continuing to develop additional ones. Also under development, but no agents released yet, include cheatgrass, Medusa head, French broom, Scotch cotton thistle, and crystalline ice plant. So some of the ones that we're doing the most work. So here's yellow star thistle. I'm sure most of you are familiar with it if you clicked on the join meeting to talk about yellow star thistle. Centaurea solstitialis, the Latin name. It's in the sunflower family, and it's native to Eurasia. It infests over 14 million acres in America and has a great economic and environmental impact. Also an impact, we have several military bases interested in this weed. It plays hell on parachutes. If you want to parachute into it, train people for parachuting, and there's a bunch of yellow star thistle out there, it can really tear up those parachutes. And also for military ground training, it makes it difficult for military bases to do their work. And here's a couple photos to help you ID the plant in the field before it's putting on flowers and sticking you with its thorns. The rosettes tend to be a bit pale, with sort of even a slight blue tinge to them, especially in the newest growth there in the center. The rosette leaves will have distinctive arrowheads that you can see at the ends of the leaves, and they're often narrower and longer than the ones you see on this particular plant. But that's a relatively distinctive feature of the yellow star thistle rosettes. When they bolt, the plants, they have these strange leaf-like keels on them along the stems. It's like a leaf growing all the way up and down the stem, which is pretty distinctive. And, again, that sort of pale color, especially when it's a younger, newly bolting plant. So now that you can ID it, get out there with your hoe and dig them up before they set seeds. In terms of the rosettes, before they develop those distinctive arrowheads and like the picture before, I don't know what to tell you. They look like a lot of other broadleaf seedlings. So maybe it has that little sort of pale color, but at that stage, they're very difficult to tell apart from other plants. So in the native range of Eurasia, there are 6 recorded subspecies of yellow star thistle, as well as a couple other related species that look pretty similar. Here, the one on the left is tocolote, which may be familiar to some of you, especially in Southern California. Centaurea is a very large genus with over 300 species in Eurasia and quite a few species that are invasive in America, as many of you are familiar with. Yellow starthistle is primarily an outcrossing species, so the genetics of its introduction to California are complicated. With a pit stop in South America that has been proposed based on genetic studies. It's likely that it first came to Chile before it came to South America or to California. Excuse me. And other introductions from other parts of Europe and Eurasia have been inferred from the genetic studies. The complicated plant genetics can also affect biocontrol. For example, I mentioned earlier that some agents may not attack all subspecies of yellow starthistle. This fly is a gall fly from Italy. Is so specific to its strain of yellow starthistle that it doesn't attack our strain here in California. Fortunately, a close relative of this fly was found, and it does attack the California variety and has been released as an agent. So here in California, yellow starthistle is fairly widespread. This map is from 1997, so if anyone knows of a more recent map, I haven't found it yet. I'd be most grateful if you have a more recent version of this map showing yellow starthistle's distribution in California. If you could send it along, I would love that. Or put a link to it in the chat. That would be fun. But as you can see, it tends to be more common in Northern California, sort of north of Santa Barbara, San Luis Obispo, and north, as I just mentioned earlier, the relative tocolote, Centaurea melitensis, tends to be more common in Southern California than yellow starthistle. But yeah, it's very widespread in Northern California and in the valleys along the coast as well. So for the biocontrol agents, this weed has, as many of you know, has been a target of biological control for many years. The first release of biocontrol agent for yellow starthistle was in 1969, which was that fly that I mentioned from Italy that never took. And then they, like I said, they found a close relative, this Urophora Cyrenaica, that did establish in California. The insects mentioned, shown here in green, are the ones that have been the most successful in terms of establishment and distribution spread. But the other ones have also spread a fair amount. One thing, it's not specifically mentioned in this slide, but I will mention it now, is that all of these insects attack the seed head of the plant, and the larvae of these insects destroy the seeds inside the flower head. So that is certainly an important stage of the plant to attack in an annual plant that relies on seeds for reproduction. It doesn't have vegetative reproduction. So they are attacking a very important part of the plant, but the plant, as many of you, if you're familiar with the plant in the field, they can make a lot of seed heads, and they keep making seed heads for several months. So so they can grow out of a certain amount of damage by some insects. And I should point out, you can see here at the bottom, one of the more successful insects was actually unintentionally introduced. I'm honestly not familiar with the whole story on that, but it was unintentionally introduced. It may have been because there was a close relative that was also worked on and introduced. But that one became very successful, even though it was unintentionally introduced. And you can also see here that the last introductions of insect biocontrol agents on YST was more than 30 years ago. So so it's still a concern based on the over 100 people that are in this webinar. This map shows where the one of the most successful agents, Eustonopus villosus, it's also known as the hairy weevil. You can see that the spots in the map are where this insect was released as a biocontrol agent, and looking at the number of spots on this map, you can pretty easily see that this insect is relatively easy to rear in the laboratory. You won't get this many releases unless you're able to crank out a lot of insects in the lab. So that's something that I'll talk about in a little bit more. But a lot of releases and a lot of success with this insect, and there's a good chance if you have yellow star thistle on your land or the land that you manage, there's a good chance that this insect is there already. And here we have a study by CDFA, who also did the rearing in that previous slide, and the releasing. This is a comparison of the distributions of the two most successful yellow star thistle biocontrol agents, showing that they complement each other in terms of the environments that they thrive in, which is a nice sort of serendipitous result, especially considering that the fly was not even intentionally released. But you can see here that one or the other of these insects is in most parts of California where we have yellow star thistle. And other studies by CDFA have tracked successful reductions in YST populations, yellow star thistle, following agent releases. Obviously, yellow star thistle has not disappeared from California, but there have been localized reductions that have been successful, and you may be familiar with some of those in your area. So here's a photo showing a location in Oregon before a release of Eustonopus, the hairy weevil. Here you can see a lot of bolting yellow star thistle in the foreground and stretching all the way through the hills there in the distance. And this land had been grazed by cattle without much effect on the yellow star thistle. But 4 years after the release of the weevil, there's a large reduction of the yellow star thistle, up into the hills. And these agents, if they establish well and they multiply well, there's no holding them back. And this was without any grazing. And some of you may be familiar with the release of the yellow star thistle rust fungus around the turn of the century. Unfortunately, it has not established well in California, and extensive monitoring was done after releases at many sites. by CDFA in the early 2000s. It needs more frequent dew in the summer than we typically see in California, except in coastal regions. And that's where the fungus has actually established in some areas on the coast. But this is an example of the target weed being able to colonize more environments than some of its natural enemies. And this one really only is establishing on the coast, unfortunately. So many of you may have also heard of the yellow star thistle rosette weevil, Ceratapion bassicorni. It's the first insect agent, well, first agent of any kind released on yellow star thistle in about 20 years, and the first insect agent in more than 30 years. It is the first agent released that attacks, or the first insect agent really, that attacks vegetative parts of the plant. It attacks the root crown of the plant. Of course, the rust is all over the plant. It was tested on 51 non-target plant species and approved for release in 2019. Rearing of this insect, as I'll talk about in the next few slides, has been a challenge. And there's two methods that have been developed, and we have a lot of work left to be done on that, and I'll talk a little bit more about that. It's been released in 3 sites in California, in Solano County, El Dorado County, and Monterey County, but it has not yet it's not yet taken hold in any of those places, and in large part because the rearing has been so challenging that we haven't been able to release more than a few dozen insects at each site. And you see here in the lower left our technician Irene, who's releasing some of her babies into the wild, and there's just at this present time not enough to release to really improve the establishment of the insect. So for the rearing, we've trained some cooperators in other states, California, Colorado, and Idaho, to rear these insects. So we have a sort of a consortium going of people trying to do our best to mass rearing this insect. And we're doing as good a job as we can, but we're not yet to the point where we can release 1,000 or thousands of insects at a time, like was the case in some of the insects I talked about earlier. Here's some photos of our mass rearing efforts. Got to grow a lot of plants. This insect is a univoltine insect, which means it has 1 generation in a year. And because of that, and it takes a relatively long time, and there's a lot of different sort of stages of its life cycle that require handling and different manipulations at different parts of the insect's life cycle. So it can be a challenge. You can't just sort of put it on some plants and stand back and watch them multiply like you can with some other insects. One effort that has been tried to increase the rearing capacity has been to create artificial diets that the larvae can feed in, and then develop into the adult stage. Unfortunately, the insects that you see here in these photos at the bottom are more or less the extent of the insects that have been successfully reared. It has worked very, very at a very low rate, but we are still plugging away with the rearing as well. And Dr. Ikju Park, UC Riverside, has been involved in this rearing effort, and he, I believe, is continuing to tweak the recipes and work on different aspects of the rearing that are relatively challenging technically, and so we're hoping that Ikju can continue to try to make progress in the rearing. In addition to the logistical challenges to rearing Ceratopion, we have also encountered biological challenges. One of these is the discovery of a male-killing Rickettsia species of bacterium that was discovered infecting our colony and distorting the sex ratios in our colonies among the consortium of insect rearing for Ceratopion. Under normal circumstances, you would expect a 50-50 male-female sex ratio. So you see the parents at the top here mate and produce 50% females and 50% males. However, when a female is infected with the Rickettsia bacterium, and the little figure of the bacteria here is just illustrative. It's not our bacterium, but it is a gram negative rod, I believe. But when the female is infected with Rickettsia and mates with an uninfected male, her male progeny die as embryos, and she produces only female progeny who all carry the Rickettsia. So it's not something that you want to find in your insect rearing colony. Probably came in with insects that were imported from Greece. That is where we get our insects, our Ceratopion, from the native range. I was just there a couple weeks ago collecting more of them. And we are currently developing DNA screening protocols to try to weed out infected individuals so that we can only work with clean, healthy weevils that will not be affected by this endosymbiont pathogen. We are also working with other biocontrol candidates for yellow Starthistle, including the dusky weevil. This is a weevil that we have imported from Bulgaria, and it has a high impact potential. Our host range testing is almost completed, and the insects, larvae eat all the seeds in the seed head that they're oviposited into. One larva per seed head, and they take out all the seed heads. But in addition, the adults, and you see here the circles at the bottom, they feed on flower buds that are developing flower buds on the plant, and that's what the adult, especially the female, feeds on to get nutrients and energy to produce her eggs that she then later oviposits in the more mature flower buds. Not quite to where they're flowering, but before they're flowering, she inserts an egg into the flower bud, and the larva eats those seeds inside the seed head. And this insect almost by definition is easier to rear than Ceratopia on past corning. But you put the insects on the plants, and they in general are pretty happy, and they start feeding and laying eggs. And you can, as long as you grow enough plants for them, they'll make a lot of offspring. So you can get over 100 offspring sometimes from a single female. Another candidate under study right now is the yellow star thistle blister mite, Cereus solstitialis. This one is we have sort of earlier stages of host range testing underway for this Eriophyid mite. For those of you not terribly familiar with Eriophyid mites, they're very, very tiny. This is one in this top photo. There's one at the end of this eyelash, just to show you the scale of the size of this mite. They're very, very tiny. They're really not, they're not visible really the naked eye. I mean, if you really squint, maybe you can see one. One reason that we have a photo with one on an eyelash is actually people who study Eriophyid mites, they often pluck their own eyelashes and glue them to the ends of little sticks to manipulate the mites under the microscope or the dissecting microscope to move them from one plant to another, for example, because eyelashes are very flexible and the mites stick to them pretty well. But that's just a sidebar for yellow star thistle. This particular mite is, as I said, we're getting in the early stages of host range testing. You can see that they can build up pretty big populations there at the top photo on the leaves. And you can have a pretty severe effect on the plant. On the lower left is a yellow star thistle plant in the field that was infested with the mite and just doesn't look very happy at all compared to a normal yellow star thistle plant. The ones on the right here in this other photo also infested with the mite and not looking happy. Unfortunately, or I don't think it's a serious barrier to release, but this mite does attack Centaurea cyanus, which is probably the closest relative of one of the closest relatives of yellow star thistle in its native range. You might know it as bachelor button. So it is a horticulturally interesting plant, but I'm not sure that attacking bachelor button would be enough to prevent this mite from being approved for a release if it doesn't attack anything else, because it's just I don't know if if big bachelor button could shut us down, but it's it's usually a plant that's put in sort of wildflower mixes that people sell. It's not really a huge industry, I don't think. So that's one that we're working on, and watch that space. So do you want to release biocontrol agents on your land for yellow star thistle or any other of your weeds? Well, you can check and see if there are any available to you, and I would you can also go out onto your land and look for them. There may be some that are already in your plants that you can sort of maybe take some yellow star seed heads that are infested with insects, and you can sort of identify which ones those might be, spread them around. You can go to other places where you know there is yellow star thistle with some of your insects and spread those around. But really just contact CDFA or your local extension, and they will help you if you want to get some biocontrol agents going. And remember what I mentioned earlier in the talk about pre-release and post-release monitoring of your land before and after you might release any biocontrol agents that are out there or available to you, and which weeds in California have available agents. Here's a list of some that have been approved for release, or plants with agents approved for release. Again, get in touch with CDFA or your local extension agents if you're interested in bringing out some agents to your land. And of course, yellow Starthistle is the one we featured here today. So with that, I'd like to again thank you for your invitation to speak here, and thank you all for your attention. And I'd be happy to try to answer your questions. I am relatively sort of new to this this target, but I actually did work on this 20 years ago with Link, but I was in between then and now have been working on other targets, and I'm back to yellow Starthistle now. So I'll be happy to address any of your questions. Thank you.
So we do have a number of questions. Hopefully, people have put their questions in the Q&A as opposed to the chat. If it's in the chat, we may miss it, but that was a fantastic presentation. We appreciate it very much.
Dr. Rector, someone's asking if you know of any plant species that outcompete yellow Starthistle.
That's not something I'm very familiar with. Julie, I don't know if you know the answer to that. Julie Julie worked on yellow Starthistle as an undergraduate student, and so I'm open to your help. But that's a really good question. I have never been asked that question. Given how invasive it is, I think it's the kind of invasive plant that it's invasive because it's sort of in areas where there's not a large amount of management taking place. I think you can manage it in terms of throwing some seeds out there and having plants outcompete it as your management. I'm not familiar with that, but I like the idea.
Yeah, and thanks for fielding it to me. I have never seen any plants that overtake yellow Starthistle, and most of my work has been in a rangeland setting. So moving on to the next question. Somebody is asking for advice about how to convince their neighbors to be responsible and take care of invasive plants on their respective properties. Do you have any good ways that neighbors can approach neighbors about this?
Well, there are certain weeds that citizens are required by law to control on their land. There's a list of those plants. I'm pretty sure at the CDFA, CDFA should have information about which noxious weeds are covered by those noxious weed statutes, which require people to kill the weed if they see it on their land. So you might, if the weeds that you're concerned about are on that list, you might inform your neighbor that we're all required to control these weeds by law. And otherwise, I would, I'm more of a carrot than a stick guy, but I you might offer to help them clean up the weeds on their land, and maybe they'll come to see the benefits. Um, but that's kind of a thorny issue, certainly. But I don't have I'm afraid I don't have any magic answers for that either. So 0 for 2.
Question about the if yellow star thistle biocontrols work on other Centaria species, specifically Tocolote, Maltastar.
Well,
If-
we typically will. Oh, sorry. Did you?
There was a second part of the question, sorry.
All right.
And if not, are there approved agents to use on these other species?
So, well, Tocolote is certainly one of the species that we test. That's a very close relative. And to my knowledge, I don't know about the historical agents that have been released. The rosette weevil does not attack Tocolote. Unfortunately, if that's a weed for some people. Back in the 60s and 70s, there was less concern about non-target plants, and so even if non-target plants were tested and found to be attacked, regulators would still approve them for release, reasoning that the weed problem was more important than the spillover might be. And so these days that's no longer the case, but maybe with some of those earlier released insects, they might have some spillover on something like Tocolote. But I would, I'm not familiar with any specific examples, but I would encourage you to, if you have Tocolote on your land, to maybe go out when the seed heads are filling in, and the insect or the plant has flowered and the flowers are starting to senesce. You might collect a bunch of Tocolote seed heads and see if you find any of those insects inside. You'll find either a little beetle or a fly larva in there, maybe.
The next question, the questioner would like to know if grazing animals, native or wildlife or domestic, tend to distribute seeds in their waste, and if there are some graziers that may be worse with this regard.
I think that that's another excellent question. It's a really great group. I think that in general, when yellow star thistle is grazed, you tend to do it before the plants are flowering and have those spines, and the spines really discourage animals from grazing on the plant, which is part of the problem. So I think in general, the livestock are not and the wildlife, to my knowledge, I don't know a lot about this, but they're not eating the seed heads, and so the grazers are getting the plants when they're younger, so they really shouldn't be spreading very well.
Good. The next question is about biocontrol agents and their success. What is the total number of agents that have been introduced, and what percentage of them have achieved a goal of significant sustained negative impact on target and with no significant impact on non-targets? And this question is not specific to a certain target species. So I guess it's more of a general overall question about biocontrol agents. And there's
Yeah, there is a catalog of, and this catalog it used to be printed out. Now it's on the internet. A catalog of weed biocontrol agents, and it is currently being managed by a woman named Rachel Winston. Earlier versions, and I believe the original version was produced by a scientist named Mick Julian from Australia. But this catalog has a list of every biocontrol agent that's ever been released on a weed to their knowledge, and information about the outcomes. Now, I don't have that at my fingertips. You could Google that biological control of weeds catalog by Rachel Winston. But off the top of my head, I believe that somewhere in the neighborhood of 30 or 40% of agents will establish on the target weeds and have an impact. And in terms of non-target effects, there's actually it's actually a very, very low number of non-target effects. I'm going to say less than 1%. There's been studies documenting. There was one recently by Suckling and Sforza documenting non-target attacks of biocontrol agents. And I'll note that, as I mentioned before, back in the day, in the mid-20th century, up until around the 80s or 90s, people would release agents. The regulators were much less strict about which agents got released, and even if they knew that they had potential for non-target attack, they would still approve them. Like I said, reasoning that the benefit from the approval or from the release would outweigh the risk of the attack on native plants. And since that time, the attitudes have changed, obviously, and people are more concerned with the welfare of native plants. So the non-target is really, especially for releases that are made these days, is very, very, very low.
Next question is about next generation research. Are you aware of any efforts to use CRISPR-Cas in IPM, invasive plant neutralization, and insect rearing?
Yes. Now, it is very cutting edge, and there are a lot of controversial aspects to using those and other types of genetic modifications, whether it's to modify an agent, a biocontrol agent, or to modify the target itself to sort of give it a suicide gene, if you will, or enable the genetic control. But this type of research is being explored. The regulation of release of that type of organism is, like I said, very controversial, and there are really very few precedents. In the plant world, no precedents that I'm aware of. Although you do have, of course, genetically modified crops, and there's data that people can get from those crops about spread of genes into other species that other than the intended one. But those tools are being explored, and I think we're a ways away from any type of release. But the regulatory issues would have to be addressed first.
If successful establishment happens at some point, would the rosette beetle be more effective than other biocontrol agents because it attacks the root crown instead of the seeds? Can it kill yellow star thistle before it sets seed?
In the field, in the native range, typically no, it's not killing the plant dead, but it is weakening plants. And the idea is that this insect can be a complement to the seed head feeding, so you have insects attacking the plant at more than one place. With the seed feeding insects, they can be competing for seed heads to lay their eggs in, and so which is a good problem to have. But with the rosette weevil, we are hoping that it will weaken plants to produce fewer seed heads, and then those fewer seed heads will be cleaned up by the seed head attacking weevil or insects.
Does grazing need to be removed for the Stenopus villosa to be effective?
I think, the weevil is out there. It's very widespread where it exists. I had that one slide showing the Eusinopus and the Achederellia, they're sort of complementary distributions in California, but the weevil's out there. So what the grazing is going to do is just give them fewer seed heads to attack. So same number of weevils, fewer seed heads, you would expect better control of the remaining yellow star thistle plants. And when you have grazing, obviously it's in a very targeted area where you take your animals and put them there. There's going to be yellow star thistle plants elsewhere nearby that the Eusinopus and other agents will be able to attack in places where you didn't put your animals, so your livestock. So they're complementary. It's an integrated type of situation.
Question about challenges faced during the research phase of biocontrol agents. When you have one in development that is having issues with bacteria or slow reproduction, why do you continue with that species as opposed to alternate ones that could be as effective?
That is an excellent question that we ask ourselves, not every day, but maybe every other day. The story of the rosette weevil is a very long and tortuous one. It was over 20 years in development. Some of that time was had to do with some data that indicated some very minor attack on safflower, which delayed things, and much more data was created to show that the insect is not a threat to safflower. But yes, could we better have a better use of our time on different research? That's definitely something we ask ourselves, and we do our best to get to an advanced stage of a biocontrol program is already like, you don't want to just give that away. So, but we do have these other candidates that I mentioned at the end of the talk that we are working on, and we're working concurrently on those with the rosette weevil, and so yeah, so it's a balancing act, and if we had half a dozen more candidates for yellow star thistle that were sort of available to work on, maybe we would put some that are more difficult a little bit more to the side, and explore some of these other ones. But at the moment, we have these two other ones that are at certain stages. So, yeah.
Are you aware of any yellow star thistle biocontrol agents that are available for purchase and release for the Southern California area?
I think the people to ask that question are in CDFA and in your neighborhood extension office. Yeah, I'm an entomologist. I go out in the field and I look for them, but and I don't have to pay for them. But yeah, I would check with CDFA and extension. They will, definitely be able to track down something better than I can tell you right now.
What percentage of a yellow star thistle field will be eliminated by the most efficient weevil? Do the weevils maintain a certain percentage of yellow star thistle to ensure their survival?
Well, I don't think they consciously do that, but I do think that it depends on a lot of factors. When you bring these weevils in from overseas, you're sort of they kind of become invasive in their own way, in that they are released from their natural enemy pressure in their home range. They have parasitoids or other things that might be attacking them. Just in the same way that yellow star thistle came here and didn't have its natural enemies attacking it, and was able to become very invasive. So the examples of the hairy weevil and the false peacock fly, they will be in almost every plant that you look at. If they're in the field, they will be in almost every plant, in almost every seed head well, maybe not every seed head, because they have a season and the plant is producing more flowers later into the season. So the insects have a season and they'll be in every seed head during that season. So part of the answer to the person's question is that the plant is actually producing more seed heads to ensure its own survival. Not that the insect is leaving some uninfested. The insect is they're greedy little critters. They want all the seed heads.
We'd like to know some advice from you how people can check their properties to see if there are biocontrol agents present. What parts of plants would they look in? And also, is there a map of release areas available to the public?
Those types of maps, again, I would lean on CDFA. They produced most of those maps. And when you're in the field, given that all the agents presently on yellow star thistle, except for the rust that I mentioned, which is on the coast. So if you live on the coast and you have a yellow star thistle problem, you can look for the rust, and it's very clear that when a plant has rust. There's little spots on it all over it. In terms of the insect agents, if you look at the seed heads, typically on the bottom portion of the seed heads is where these female insects will lay their eggs. And so you might see a little hole in the bottom of a seed head, which would be a good indication that you have an insect. But some of them can be on the side, some of them, but typically on the bottom or the side of the seed head, if you carefully, and they're very spiny, so use gloves and everything. But if you look closely at these seed heads, particularly at the time when they're really sort of blooming the flowers are in full bloom and not drying out yet, I think that's a good time to look for them.
Before we go on to the next question, we have a few people who have asked if this presentation will be made available. And it is being recorded. It will be posted likely next week on the UC ANR California Invasive Species Action Week Lunchtime Talks webpage. Randall, do you have anything to add to that?
No, hopefully, I mean, I need to go in and do some very minimal editing and clean up the transcript, but it most likely will be live on that site tomorrow.
Somebody starts the next question with an apology, and in case they didn't hear you say this, Dr. Rector, but they'd like to know where yellow star thistle is native from and how did it get into California?
Yes, I did cover a little bit of that earlier in the talk, but I'm happy to recapitulate. The plant is native to Eurasia, which obviously is a big place, but typically from Mediterranean areas of California, of course, has a Mediterranean climate. There's parts of California, there's Mediterranean climates in South America and other parts of Australia, other parts of the world. So yellow star thistle is from the Mediterranean, like the Mediterranean area. And both north and south, sort of the Mediterranean in Europe, Southern Europe and Northern Africa into Western Asia. As I mentioned earlier, it's a complicated story of the introduction, starting in Europe and then first to Chile. Has been inferred from genetic analyses, and then from Chile to California. But other parts of America, particularly in the West, have also received yellow star thistle from parts of Eastern Europe, like in the Balkan region, where I was just a couple weeks ago, and Turkey, Uzbekistan, and that type of area.
The next commenter notes that it seems that overgrazed land or highly disturbed land makes it easier for yellow star to establish. Is there a land management component that you've seen with where populations are?
Well, that's a very common characteristic of many invasive weeds, because native plants, are adapted to grow in their native habitat, which is not disturbed. And some native plants will, grow well in disturbed areas, like crabgrass or something like that. But yeah, it's a characteristic that yellow star thistle shares with many invasive plants, and so it's something to think about in the land that you manage. Areas that are disturbed, you want to be vigilant about what types of weeds might come in if you disturb certain parts of your land.
Nick, how does government, and at what level of government are requirements for noxious weed removal established?
That is a state by state issue. I'm not sure. I'm really not sure about federal. I mean, I know that there are some plants that are federally restricted, like I suppose coca plant, or cannabis. I think was recently removed from the highest list, but I don't know. It's generally a state by state issue. So your state government.
Is prescribed fire antagonistic to the use of biocontrol agents or can or should they be coordinated?
I think they can be coordinated. I think that again, prescribed fire is a lot like grazing in the sense that you do it in a very specific spot, and you know, you put your livestock in this one place and you don't want them going everywhere else. You put the fire in one place and obviously you don't want it going anywhere else. So outside of that treatment area, you're going to have yellow star thistle plants or other weeds that have biocontrol agents on them. You're going to have them outside that area anyway, and so the insects and the agents are still going to be able to reproduce in those non-treated areas, whether it's by grazing or prescribed fire. So yes, certainly those two things can live together and complement each other.
Have you seen any cross-pollination between yellow star thistle, spotted knapweed, and if the biocontrols work on this hybrid?
I am not aware of that hybrid. It would surprise me if that were a possible hybridization, just because I don't think they're terribly closely related. I think you'd be more likely to see a tocolote hybrid with yellow star thistle, but we've had people send us plants that they thought might be hybrids between yellow star thistle and tocolote, but we don't really know if that has ever occurred, and you know, it's something where if you have a really unusual weather year, you might see tocolote that looks different than you're used to seeing it, maybe bigger or smaller, and it's not necessarily because it hybridized with anything. And the same for knapweed or yellow star thistle.
So there's a follow-up comment to this last question by someone who did find a yellow star spotted knapweed hybrid, and it was confirmed by CDFA, their plant pest lab. So maybe it does occur.
Sure.
Yeah. And we're at our last question, which is like the elephant in the room, because the questioner wants to know about the worst outcome that has happened from a biocontrol release that you know of, where a non-target impact was significant.
Well, you know, there are some examples. For example, one example that happened here in California. Well, maybe not exactly. So there were a lot of thistles, like musk thistle or milk thistle, and there were several different thistles that were subject of biocontrol programs in the 60s and 70s, and there was a weevil called Rhinocilus conicus that was released, and in the studies of this weevil before it was released, they observed that the weevil would attack some American native thistles. But the regulators decided that negative did not outweigh the positive of the thistle control by the weevil. So the regulators went ahead with allowing the release of this insect. And so eventually in the 80s and 90s, some people noticed that some Native American thistles, there were some particular examples in the Midwest, the Great Plains area, where some relatively rare native thistles were having these weevils eat them or eat their seeds. So again, a case where the science actually predicted that this would happen, but the regulators decided it wasn't important enough. So these days that's really changed, and people tend to be a lot more concerned about the native flora being attacked. And actually, one interesting aspect of that particular story is that looking back at some of the weevils that were released or collected in Europe for release in the US, especially some that were collected and released in California, there's some question as to whether the weevil that was released was actually Rhinocillus conicus. It might actually be a different species. So not only did they permit the release of some insect that was not very host specific, but they might not have even released only that one insect. They might have accidentally released some others. There's some other examples in the West. The cinnabar moth is a famous example, and also there's an example of the cactoblastis moth attacking Opuntia cactuses, which have been very invasive in different parts of the world, and the moth is spreading to places where there are more rare examples of relatives of the cactus that weren't tested because they weren't in the release area, but the moth had spread after that into other countries. So there are a few examples of those things happening, but they're compared to the overall number of really hundreds of different species released for control. And you can see those, and thanks to Randall for putting the links to some of those databases, and also other people put links in the chat. The frequency of that sort of non-target effect is very rare.
Good news.
If it wasn't, we'd probably not be doing this. So. So thank you for all those questions, especially educating me about things like spotted knapweed hybrids and things that I didn't know about. I wish I could have answered more of the questions better than I did.
You did a great job. Thank you very much. We've gotten numerous positive comments in the chat and elsewhere. So thank you for staying and answering all those questions. You went way beyond the call of duty. And thanks to everybody who has stayed on. And I would once again say, please do complete the questionnaire that is listed in the chat if you haven't already done so. And with that, I will end the call.
Thank you, everyone. Thank you, Dr. Rector.
You're welcome.